So today we're going to be doing a lecture on enzymes and how they work and whatnot. So the first thing we're going to do is define enzymes. So enzymes, what they basically are proteins. And so if you remember, proteins are made out of amino acids. So this is our basic structure of an amino acid. So these R groups can be pretty much anything you want. You know, they can be OH, they can be an H, you know, they can be just a CH3 group. Really, they can be a lot of different things. So this is just a basic structure of a dipeptide, and this would be our peptide bond right there. So that's all we really need to know in terms of what proteins are and what enzymes are exactly. So let's move on to their structure. So their structure is broken down into primary, secondary, tertiary, and quaternary. So primary is just based on your amino acid sequence. So say you had like a serine, then a glycine, and blah blah blah, whatever it may be. The whole peptide chain, the whole amino acid chain, that would be your primary structure. So secondary, what would that be? So secondary is how it actually folds. So the intramolecular forces that may interact here, but only the intramolecular force of hydrogen bonding. So hydrogen bonding has to look something like this. So that would be hydrogen bonding. It's a weak intermolecular force but it's the strongest of the weak. This hydrogen bonding within the amino acid structure would cause something, two different things. We have this and we have this. So this is called our alpha helix, and this is our beta pleated sheets. So depending on how these amino acids interact with each other, depending on these hydrogen bonds and how close they are together, they can form into these different secondary structures, okay? So we can think of them in 3D, but we don't really consider them 3D quite yet, okay? So tertiary structure is really where we see this folding of the secondary structure, and that's where we really can see that it is 3D now. So we can imagine we have something like this, and it's like a round little ball, and that what we normally see in these enzyme diagrams with all the different bondings, and we can rotate it around, that would be the tertiary structure. So if you can imagine, this is 3D, and if you can imagine, there'd be a bunch of different beta pleated sheets and alpha helixes surrounding it and also within. So you would see it all around there, and so these actually have more than just hydrogen bonding as the intramolecular force. They have hydrophobic interactions. They also have obviously hydrogen bonding, ionic bonding, disulfide bonds. So it has a lot more intramolecular forces. Okay, so that's what a tertiary structure would look like. So what would quaternary structure look like? Well, so this was one entire amino acid chain, just one chain, no breaks. So if we had two of them together, so this was one chain of amino acids, so you had N all the way to C, so that's the first chain, and then another N to C, that's the second chain. And if they're put together like that, probably through disulfide bridges, hydrogen bonding, hydrophobic interactions, that would be a quaternary structure. So it's the combination of two or more tertiary enzymes or proteins, whatever you want to call them. We'll just call them two or more tertiary enzymes together. Right? So how do enzymes work? So this is just a very, very basic definition or drawing of how it works. For the MCAT, this is all you really need to know. So imagine if this was our little enzyme right here. Right? This would be considered the active site. Okay? So this is where everything happens. All the reactants, all the substrates will interact in this active site. So say our final product is we want to combine A and B, right? And so this is our reactants and this is our products, right? So we have A and we have B and we have them all floating around. But we need this particular bond to be made. We need let's just call these 2 points, point 1 and point 2, they have to attach, and that's where the bond has to be made. The chances of them interacting with each other are very, very unlikely. If you can imagine, there's so many other possibilities for these 2 points to meet that the chances of them meeting directly are very unlikely. But now imagine that this enzyme grabs A in the back and has that point sticking out. Okay? And it cannot fit any other way. And now imagine you have B comes around, it's floating around, I have a lot of B's, and they come right there. And so now you have both of the points that are supposed to bond together facing each other. And now you have a much higher chance of having that bond to form, and they get put out, and now you have this product. So that's really how enzymes work. They work in the active site, that's the first thing that's important, and they increase the likelihood of positive interaction, so positive bonds to be made, pretty much, or bonds to be broken. Enzymes can work both ways, or transferred as well. So regulation, there's a bunch of different types of regulations in terms of enzymes, but for the MCAT we really only need to look at this one right here. So it's allosteric inhibition. Okay, so if we have our same enzyme right here, this was the active site. So now let's imagine, so if you can imagine maybe an indentation here, we'll call this the allosteric site. Alright, so this allosteric site is where some type of allosteric inhibitor, let's just call this A. So this allosteric inhibitor will come in, bind to this point right here, and it'll have some type of conformational change. So when A is bound, now it had some type of conformational change that now all these reactants, they can't get in, right? All our B's, our C's, whatever had to bond in here, they can't get in here. They can't get into this point. So now no bonds can be made. The enzyme is inhibited. Okay. So pretty much that's how it works. It's just that we need to know that allosteric is different from the active site. Alright, so similar to allosteric inhibition, we have something called negative feedback. So negative feedback, the most basic would just be something like this. So say A is somehow transformed to B by enzyme 1. Okay? Negative feedback would say that, hey, we have this B, this product that we formed, now it's going to inhibit the reaction from A to B. Okay? So that means when we have an increased amount of B, the reaction A to B will be decreased. That enzyme activity, so E1 will be decreased. The enzyme activity of E1 will be decreased because now we have B, and it's a negative inhibitor or allosteric inhibitor, negative allosteric inhibitor for the transformation of A to B. And we can just see how this works. So say we have a typical case, A goes into this active site and it changes so that now this A turns into a B. Okay, so this B comes out and say it comes all the way to the allosteric site, right? And now it binds to the allosteric site. Okay, now there's some type of conformational change. Okay, so B is attached, some kind of conformational change, and now so there's an inhibition of the change from A to B. A can no longer get into this active site, so the enzyme cannot do what it has to do and change to B. Okay? So that's pretty much what a negative inhibition would be. Obviously we could have a much bigger, and this is generally what would happen, not just this simple case like I showed, where this may inhibit that, that would go back and inhibit that, and we see this, and this is something we need to know for much later, is hormones. Hormones are very important. If you ever get a question where they ask you what type of inhibition are hormones, and it's this negative feedback inhibition. If you had to guess, it's probably going to be that.